Chapter 1. The Scissors They Did Not See Coming. For most of modern history, the masses believed biology was untouchable, fixed, and governed by fate rather than design. Genes were destiny, mutations were accidents, and evolution was a slow burn measured only in centuries.
The genes I turn on and off are determined by the life that I live.
Then CRISPR arrived, quietly, clinically, without cinematic explosions or press conference arrogance. While the masses were distracted by surface-level biotech headlines and miracle cure hype, the elite noticed something more unsettling happening. Life was no longer just observed, cataloged, and treated. It was being edited. CRISPR did not introduce genetic manipulation. That idea existed for decades inside specialized labs. What it did was break the barrier of complexity, turning gene editing from elite institutional science into an accessible biological tool. At its core, CRISPR is a molecular search and edit system, guided by RNA, precise enough to find a single typo inside billions of genetic letters. It cuts, removes, or replaces DNA with an efficiency that feels less biological and more computational. This was not a medical advancement alone. This was a paradigm shift. The masses saw another science headline, complex, distant, and easy to scroll past. The elite recognized a meta moment, the day biology started behaving like software. CRISPR did not ask for permission. It rewrote the rules. Chapter 2. How bacteria wrote the first draft. Every powerful technology has origins that sound almost mythical in hindsight. CRISPR began not in humans, but in bacteria fighting for survival in an invisible war. For billions of years, bacteria faced relentless attacks from viruses called bacteriophages, microscopic invaders programmed to hijack and destroy. CRISPR was their memory system, a biological archive of past attacks, stored like mugshots inside their DNA. When a familiar virus returned, the bacteria responded instantly, slicing the invader's genetic code with surgical precision. Nature built gene editing long before humans gave it a name. Scientists didn't invent CRISPR, they decoded it. And once understood, its potential exploded outward. Suddenly, diseases caused by single gene mutations were no longer abstract tragedies. They were engineering problems waiting for a solution. The masses heard terms like gene therapy and imagined science fiction timelines far beyond their lifetime. The elite understood the subtext. This was happening now, quietly, efficiently, and without consensus. CRISPR was not futuristic. It was already here. Don't follow the crowd. Follow the aura. Chapter 3. Editing errors. Healing humans.
The FDA just approved the first ever CRISPR-based therapy, a medicine called Casgevy. Both sickle cell disease and beta thalassemia. These diseases affect hundreds of thousands of people, mostly of African and Asian descent. Casgevy can be used to treat both conditions by using CRISPR to edit a gene in a patient's own blood stem cells so they resume production of fetal hemoglobin.
Medical science tends to move cautiously, but CRISPR forces speed into systems designed for restraint. Inherited disorders like sickle cell disease, cystic fibrosis, and muscular dystrophy suddenly became editable errors, not lifelong sentences. In recent years, real patients have undergone CRISPR-based treatments, with measurable improvements, not promises. These are not speculative trials anymore. They are lived outcomes recorded in controlled environments. The process is deceptively simple to describe and extraordinarily complex to execute. Cells are extracted, edited, verified, then reintroduced, corrected code running inside a human body. Cancer research shifted as well, using CRISPR to reprogram immune cells to recognize and destroy tumors more effectively. This is not medicine reacting to disease. It is medicine redesigning response itself. Yet here the tone changes. Because every edit carries risk, off-target cuts, unintended consequences, and ethical gravity. The masses ask whether this will cure everything. The elite ask who gets access first and who waits. CRISPR exposes inequality faster than it solves disease. Don't follow the crowd. Follow the aura. Chapter 4. The Ethical Fault Line. This is where the conversation tightens and discomfort becomes unavoidable. CRISPR does not stop at healing. It opens the door to enhancement.
This morning, the little boy we've been following for months who received the first of its kind gene therapy for a potentially fatal disease is marking major developmental milestones, set to spend his first Christmas at home. Now 16 months old, KJ Muldoon recently took his first steps.
It's all been a miracle. That's the only way to describe it.
Days after he was born, KJ was diagnosed with a rare genetic disorder which causes ammonia to build up in the blood and can lead to brain injury. Earlier this year, he became the first patient in the world to receive a personalized gene editing therapy called Crispr.
In 2018, global shockwaves followed when gene-edited babies were announced, edited before birth, without global consensus or ethical alignment.
A line has been crossed that should not have been crossed.
It's very disturbing, it's inappropriate.
Oh, this is huge.
The reaction was immediate, unified, and fearful. Because once edits enter the germline, they don't disappear. They propagate forward, rewritten instructions passed to generations that never consented. The technology itself is neutral. Its application is not. The masses reduce this debate to extremes: miracle cures versus dystopian futures. The elite recognize the harder truth: governance lags innovation every single time. CRISPR is moving faster than policy, culture, and moral frameworks can adapt. This is not a bug. This is the pattern. We are editing life inside systems built for caution, using incentives designed for speed. That tension does not resolve itself. Don't follow the crowd. Follow the aura. Chapter 5: Agriculture, Climate, and Silent Power. Human health is only a fraction of CRISPR's reach, and arguably not its most disruptive arena. Food systems, climate resilience, and ecological control sit quietly beneath mainstream conversation. CRISPR-edited crops can resist disease, survive drought, and require fewer chemical interventions. This reduces cost, environmental damage, and dependency on unpredictable climate patterns. Livestock research explores disease resistance that could prevent future pandemics before they jump species. This is biology acting as infrastructure, not treatment. The meta-implication is unsettling but clear. Food no longer has to adapt slowly to nature. Nature can be coded to stabilize food. The masses debates labeling and fear modification. The elite track supply chains, resilience curves, and population pressure. CRISPR is already shaping what survives the next century. Chapter 6: The future is editable. CRISPR is still young version 1 of a tool that will refine itself rapidly. New variants improve precision, reduce errors, and expand control. But technology never pauses for philosophy to catch up. It moves, iterates, and normalizes before society finishes arguing. The masses will eventually accept gene editing the same way they accepted vaccines, smartphones, and artificial intelligence. Gradually, reluctantly, and only after benefits outweigh fear. The elite are already positioning, regulating, investing, and deciding boundaries behind closed doors. CRISPR is not about playing God. It is about acknowledging that humans have always altered biology, only now with intention and speed. This is not the end of nature. It is the beginning of authorship. And the final divergence is simple. Some will react when the future arrives. Others will shape it while it's still writable. Don't follow the crowd. Follow the aura.